A T-cell receptor recognizes antigen in the form of a complex of a foreign peptide bound to an MHC molecule. Antigen recognition by T-cell receptors clearly differs from recognition by B-cell receptors and antibodies. Antigen recognition by B cells involves direct binding of immunoglobulin to the intact antigen, and antibodies typically bind to the surface of protein antigens contacting amino acids that are discontinuous in the primary structure but are brought together in the folded protein. T cells, on the other hand, were found to respond to short, contiguous amino acid sequences in proteins. These sequences were often buried within the native structure of the protein and thus could not be recognized directly by T-cell receptors unless some unfolding of the protein antigen and its processing into peptide fragments had occurred. The nature of the antigen recognized by T-cells became clear with the realization that the peptides that stimulate T-cells are recognized only when bound to an MHC molecule. These cell surface glycoproteins are encoded by genes within the major histocompatibility complex, MHC. The ligand recognized by the T cell is thus a complex of peptide and MHC molecule. The evidence for involvement of the MHC in T cell recognition of antigen was at first indirect, but it has recently been proved conclusively by stimulating T cells with purified peptide MHC complexes. The T cell receptor interacts with this ligand by making contacts with both the MHC molecule and the antigen peptide. And that's a T cell receptor recognizes antigen in the form of a complex of a foreign peptide bound to an MHC molecule. Have a great day.